Our group was in charge of the research on measuring gut contractility. The members of our group are me, Linda, Arshad, and Muhammad, under the supervision of Dr. Karen Yurey and Dr. Tibor Docha. This is an outline that represents what we're going to discuss in our presentation. We'll discuss the background, methodology, data analysis, statistics, evaluation, and conclusion. To begin, let's start with the background. I'd like to mention the disease background of our experiments, the reason why the experiments were performed, and that is ileus. What is ileus? Ileus is a condition where one's intestinal motility is declined due to reasons such as gut manipulation or trauma, but not due to a mechanical intestinal block. This condition is commonly seen in general surgery patients as well as in trauma patients. In fact, it was reported as one of the most common reasons for hospital readmission. However, despite the vast complications that it infers on its patients' comfort and health, the pathophysiology of this condition has not yet been completely uncovered. This can be attributed to its very complex development and the number of factors that contribute to its development as well, including inflammation and edema. However, with that said, there is indeed evidence that points to a chemokine known as CXCL1 playing a major role in its development. So as I mentioned, among the main contributors to ileus are inflammation and edema, which have been found to lead to ileus through multiple pathways. Inflammation has been seen to reduce intestinal motility by interfering with the enteric nervous system, a division of the autonomous nervous system present in the gastrointestinal tract. More importantly, though, inflammation can lead to the secretion of a specific cytokine known as CXCL1, which is evidence to decrease intestinal motility. Inflammation can also lead to edema. Now, edema, even in the absence of inflammation, is seen to decrease myosin light chain phosphorylation, and this is required for smooth muscle contraction. So, this eventually leads to a decrease in gut motility. It can also mechanically stimulate some resident macrophages lying within the intestinal smooth muscle to also release our targeted cytokine, which is CXCL1. This, of course, will lead to a decrease in the intestinal motility as well. Hence, we can concur that ileus is likely to be induced by inflammatory injury to the intestinal smooth muscle layers. We previously mentioned CXCL1, but what exactly is it? Well, CXCL1 is a chemokine belonging to the CXC family. This chemokine has been found to be released by resident macrophages of the intestine's smooth muscles upon intestinal wall stress. Listed here are some indicators pointing to CXCL1 as a main mediator of ileus. These include the increased CXCL1 levels in mechanically stimulated intestinal smooth muscles of ileus-induced mice, as well as increased levels in severely injured trauma patients post-hospitalization and in trauma patients who ended up developing ileus. Both of these findings were uncovered by Dr. Yurey et al. Another thing to note about CXCL1 is that it activates a CXC receptor, whose activation leads to a downstream of different pathways that could potentially lead to ileus. Now, a major issue with treating ileus is that the currently available medications target the enteric nervous system. However, as the evidence points, ileus is most likely caused by smooth muscle dysfunctionality. So then these medications are practically ineffective. So we must set out to identify a new target. Here is a visual representation of the proposed molecular mechanisms behind ileus. First, we can see that mechanical signals and P21 activated kinase both lead to the activation of certain transcription factors which lead to the production of CXCL1. Then CXCL1 will bind to its receptor CXCR, which is a G protein-coupled receptor, which upon ligand binding will lead to a cascade of intracellular signaling pathways through the activation of its G protein. CXCR can lead to the activation of phospholipase C-beta, which will produce second messengers inositol triphosphate and diacylglycerol, leading to the activation of protein kinase C. Another pathway is through the activation of the G protein monomer, RAS, and the RAF of the MAP kinase pathway. However, our target pathway is that leading to the activation of AKT, otherwise referred to as protein kinase B, through phosphoinositide 3 kinase. Now, a very interesting observation is that NF-kappa beta, which can upregulate CXCL1, is actually increased in edematous intestinal smooth muscle, resulting in decreased intestinal motility. So, this transcription factor may upregulate CXCL1 in response to increased stretch. As mentioned, our pathway of focus is the AKT pathway. Could AKT be the main player causing the decreased intestinal contractility seen in ileus? To investigate this, a series of experiments were conducted where mice intestines were suspended in different mediums including CXCL1 and AKT inhibitor. What we want to see is the increased contraction in intestines that were exposed to CXCL1 and AKT inhibitor together as compared to the contraction when just using CXCL1. If this is observed, then it is an indication that AKT can indeed be responsible for the symptoms correlated with ileus. Now, I will leave Arshad to explain the methodology.
Firstly, in our experiment, we anesthetize the mice using isoflurane and we make an abdominal incision and remove approximately 1 centimeter of the intestine and transfer it to a petri dish containing Kerb's buffer. Next, we make a split knot and prepare the intestine and load it onto our experimental apparatus, which consists of 4 chambers labeled 1 to 4. The first chamber will be used as a standard and chambers 2 to 4 will be used for our experiment. We adjust the silk such that the intestinal contractility can be measured and recorded on our computer. Next, we leave it for 30 minutes to reach equilibrium. Then we take baseline 30 minutes after the equilibrium has been achieved, and then we add AKT to channel 2 and 4. Next, we add CXCL1 to channels 3 and 4, such that channels 2 have AKT1, channel 3 has CXCL1, and channel 4 has AKT and CXCL1 together. Then, we add carbachol in increasing concentrations from 10 to the power minus 8 to 10 to the power minus 3 micromolar concentrations.
So now that we've done the experiment, let's analyze the data. Presented to you is the entire graphical outcome of the experiment, from start to finish. So notice how we have 4 distinct channels. Channel 1 features the MSO and a buffer. This represents the full control group. This is our reference, which we will use to compare the effects of different treatments. Channel 2 features the AKT inhibitor treatment followed by the buffer. This shows the effect of the AKT inhibitor on contractility. Channel 3 features the MSO followed by CXCL1. This represents the effect of CXCL1 on contractility. And lastly, channel 4 features the AKT inhibitor followed by CXCL1, representing the effect of both AKT inhibitor and CXCL1 on contractility. The biggest feature of these graphs is the oscillations. So if I take these graphs and I stretch them, in this case, you will notice there's a periodic up and down pattern. These oscillations represent the contractility of the distal small intestine of the mouse. So, all channels were equilibrium for over 30 minutes and then baselined with either DMSO or an AKT inhibitor before other treatments were added. We could really divide this entire graphical outcome into 2 main parts. We have the spontaneous contractility and agonist-induced contractility. The spontaneous contractility refers to the natural contractility of the intestine. We also added increasing concentrations of carbachol at 5-minute intervals. Carbachol is an agonist to the acetylcholine receptor, so it is a classical stimulator of contractility. This effect is valid up until 10 to the power of negative 4 concentration, and beyond that, the contractility decreases again. Linda will elaborate more on this shortly. So the part of the experiment with carbachol treatments refers to the agonist-induced contractility, which we can see here. And notice how after each treatment, each channel has its own kind of graphical change, indicated by the arrows at the bottom. For now, I want to focus your attention on channel 1 and channel 3. So, in channel 1, the buffer caused no significant change in spontaneous contractility here. When the carbachol treatments were added, however, we can see an overall increase in contractility, which is to be expected since it goes through cholinergic receptors and is involved in muscle contraction. However, we must say that we expected a way larger increase in agonist-induced contractility, and we assume that this is because the tissue might have died or was harmed in the process, which decreases the contractile effect. But if we compare it with CXCL1, which was baselined with the MSO as well, we can see that CXCL1 did not affect the spontaneous contractility. So notice how after we added the CXCL1, there wasn't a big difference in contractility. However, what CXCL1 did was induce a significant decrease in agonist-induced contractility compared to the buffer treatment. This means it's relaxing the smooth muscles of the gut, and we can see an overall decline pattern here. Let's consider the AKT inhibitor now. But what's the role of AKT in the first place? So we know that CXCR2 is a receptor for CXCL1. When CXCL1 binds, it triggers 3 signaling pathways. Our current focus is on the AKT pathway, which ultimately leads to decreased intestinal contractility. Our hypothesis is that if AKT is involved in decreasing intestinal contractility, then using an AKT inhibitor should act as a CXCR2 antagonist, which would then prevent this decreased intestinal contractility. So, by comparing channel 1 and channel 2, we can notice that the treatment with the AKT inhibitor did not have an effect on both spontaneous contractility nor agonist-induced contractility. So there's no significant change in the amplitude or tone in both phases, the spontaneous and the agonist-induced. But what's really interesting is that if we look at channel 4, where we used AKT inhibitor followed by CXCL1, the AKT inhibitor seemed to inhibit the decreased agonist-induced contractile activity induced by CXCL1. We can see this by the increased contractility trend upon the addition of the carbachol treatments. So AKT doesn't affect spontaneous and agonist-induced contractility by itself, but it seems to affect the CXCL1 pathway to some extent, since it inhibited the effect of CXCL1 when it was used with it.
Now, after we've analyzed our data and to make sense of our results, we are going to look at the statistics. To make sense of our results, the following values were measured. First, we measured the integral, which is the area that was indicated under this curve over here. We also measured integral from minimum, which is the area that was calculated under the curve but from the minimum point of the curve. Why did we do this? Why did we use integral and integral from minimum? Well, integral from minimum was used to exclude the change in tone from the integral values. So integral from minimum ignores changes in tone. And this is what tone represents. We also measured amplitude, which is the difference between the highest point and the lowest point on one curve. We also measured frequency, which is a number of contractions within a certain time period, so second within a second. And this is because intestinal contractility is measured in terms of oscillations, as Muhammad mentioned. So let's look at the actions of AKT inhibitor on spontaneous contractility. Meaning the contractility of the intestine when it is not stimulated by an agonist. So it's intrinsic contractile activity. These values were the calculated averages of integral, integral from minimum, frequency, amplitude, and tone that were obtained from the channels containing the buffer, so the channels 1 and 2, and then from those containing AKT inhibitor, so those were channels 3 and 4, both in the absence and in the presence of CXCL1. Overall, we can see that AKT did have notable results. First to note is the increase in the overall intestinal contractility in the presence of AKT inhibitor, as well as the increase in the amplitude of these contractions. We can also mention a decrease in the frequency of the contractions when we use AKT inhibitor, and a slight decrease in tone as well. Next, let's compare the contractility of the groups in the presence of carbachol, so in the presence of the agonist. So we can observe here a carbachol dose response curve to measure the agonist-induced contractile activity of the intestines. Remember that the carbachol was added in increase in concentrations from 10 to the power of minus 8 to 10 to the power of minus 3. But after a 10 to the power of minus 4 carbachol concentration, we could see that the intestinal contractility actually decreased. And this is because the maximum contractility of the intestines was at 10 to the power of negative 4. So this is the maximum that the intestines can contract. Now, looking at this graph, the key points to note are, first, the decreased intestinal contractility in the presence of just CXCL1. And this was expected. Secondly, and this is what the experiments actually sought to confirm, is the presence of both AKT inhibitor and CXCL1 serve to increase intestinal contractility as compared to the contractility with just CXCL1, and this is this pink line over here. This indicates that AKT is a possible cause of the decreased intestinal motility that we see in ileus. However, something that we did not expect was that when we use just AKT inhibitor on its own, a positive trend is observed. So as you can see indicated by this blue line here, the contractility with just AKT inhibitor is actually higher than that of the buffer solution, which is this red line over here. So all of these combined results are an indication that although AKT inhibitor seemed to prevent the decrease in contractility that would have occurred if just CXCL1 was used, since AKT inhibitor seems to have its own positive effect on contractility, then these results are not very conclusive. So, as Muhammad mentioned previously, something that we noticed is the decreased contractility that was observed in the first channel, the controlled channel per se. We suspect that this stems from our usage of a possibly compromised intestinal tissue in one of the experiments, which did not respond appropriately to the carbachol dosage. So this is what we wanted our graphs to actually look like. This is what we hoped it would look like if AKT was truly the causative agent of ileus. You can see here that the highest contractility should have been exhibited by the intestines placed in the buffer solution, followed by those placed in AKT inhibitor, and then the one in both CXCL1 and AKT inhibitor. However, as we've seen previously, our results showed quite different results, again, due to AKT inhibitor's own effect on contractility and due to the compromised intestines. Now we look at the comparisons of the effect of AKT inhibitor on contractility and on CXCL1 activity. And here we're talking about spontaneous contractility, so in the absence of the agonist carbachol. On this first slide, we'll examine the integral and the integral from minimum. Surprisingly, when it comes to the integral, the presence of AKT inhibitor actually managed to decrease the contractility. In fact, there was a 5% decrease. However, in both cases, we can see that CXCL1 increased these values by around 1%. Next is the integral from minimum. So generally, the same consensus is present, where AKT inhibitor managed to decrease the values, and CXCL1 also decreased the values here. In the absence of AKT inhibitor, it decreased the contractility values by 4.52%, where in the presence of AKT inhibitor by 7.9%. In the next slide here, we'll move on to discuss the other aspects of intestinal mobility, the amplitude, the tone, and the frequency. First, we can see that there were no vast changes when it came to the amplitude, although CXCL1 managed to decrease said amplitude by approximately the same amount in both the presence and the absence of AKT inhibitor. But of note is that AKT inhibitor alone also managed to decrease the amplitude of contractions as well. Next, we can see that here, in both the absence and the presence of AKT inhibitor, CXCL1 increased tone by 2.16%, but when AKT inhibitor was used with CXCL1, the increase was of less significance, so it was just a 1.25% increase. And finally, when it comes to frequency, with the absence of AKT inhibitor, CXCL1 decreased the frequency of contractions by 0.76%, but when AKT inhibitor was added, the frequencies were approximately the same as well, with a very slight increase caused by CXCL1. Although in cases where more than two groups are being compared, an ANOVA test is usually performed, here we performed a t-test as to make quick comparisons and obtain a rough estimate of the degree of change. So, according to the t-test results that we can see here, there aren't any significant changes in contractility in all channels. However, repeating the experiments more times could get us closer to confirming their results.
So what are our conclusions from this experiment? From our graphs, we could observe that CXCL1 decreased agonist-induced contractility, but had little effect on spontaneous contractility. And the AKT inhibitor countered the effect of CXCL1 on gut contractility, so we assume it plays a role in the CXCL1 pathway. However, according to our calculations, these effects were not statistically significant. Lastly, the role of the AKT inhibitor as a regulatory factor by itself remains unclear. Since we investigated by itself, it did not significantly change the spontaneous nor agonist-induced contractility. So we can't make any strict conclusions at the moment, but points of improvement for follow-up experiments can be made, and these include ensuring the use of live tissue, and this we can do by decreasing the time from when we surgically remove the intestines until we put them in the setting device. A complete unharmed tissue will give us more precise data. And since the effect of the AKT inhibitor by itself remains unclear, repeating the experiment several times will provide a comprehensive view of the role of AKT. Repeating the experiments will also provide us with more data, which could bring us closer to the true average of, for example, contractility and allow us to establish statistical significance. So why are experiments important in the first place? So, chemokines like CXL1 are upregulated in diseases like ileus, which in our case decreases intestinal contractility. So if CXCL1 significantly decreases intestinal contractile activity, a potential counter is to use a CXCL1 receptor antagonist to help increase the contractility again, or an inhibitor that will inhibit signaling proteins activated by the signal propagation from the CXCL1 receptor. If AKT inhibitors truly did counter the effect of CXCL1, then it has huge drug potential in treating gastrointestinal diseases characterized by decreased contractile activity. And in a clinical context, this is relevant in the case of abdominal surgery patients who experience decreased gut contractility following their surgery. And to conclude our presentation, we want to say a big thank you to Dr. Karen and Dr. Tibor, as well as their very kind PhD students in their lab for guiding us for the past few months. We are very grateful for the exposure we received in the research environment, as well as the research skills and the clinical knowledge we accumulated throughout the course. And here are our references. Thank you.